The state of Texas continues to be home to some of the world's brightest minds and groundbreaking research. The Academy of Medicine, Engineering, and Science of Texas is dedicated to recognizing the rising stars of research in the Lone Star State. Tonight, we proudly present the recipients of the 2009 Edith and Peter O'Donnell Awards.
Right, let's take a look at those. And maybe pull a control up so we can compare.
Whether he's in the lab leading a team of scientists manipulating and splicing genes, or in the hospital comforting and caring for disabled children, Dr. Brendan Lee is always moving toward the treatment and cure of rare diseases. A native of Hong Kong, Brendan moved with his family to New York when he was 6 years old. His parents were already pushing him toward a career in medicine.
I fell in love with it. Both the medicines part, which is really to deliver care and to try to impact positively patients' lives, as well as the science part, which is to try to understand what's going on in our patients.
Brendan's curiosity led him to genetics in the late 80s, as scientists were just starting to understand how DNA works. Brendan came to the Baylor College of Medicine for postdoctoral training.
Another important signaling protein called Notch is affected. And here you see almost all of the bones, all the cartilage in the skeleton is gone.
Now he leads a team of researchers in Baylor's Department of Molecular and Human Genetics. Initially, Brendan focused on diseases of the bones and connective tissues.
We identified many of the first genes that cause human skeletal dysplasias. Chondrodysplasias typically are thought to be dwarfing conditions. Marfan syndrome associated with problems with your blood vessel, associated with problems in growth also.
In 2006, Brendan and his team pinpointed the gene responsible for a unique form of brittle bone disease. This mutated gene causes bone cells to produce abnormal collagen, which reduces bone strength. Armed with this information, Brendan's team has found ways to fight the disease and help his young patients.
You'd see them in the hospital on a pillow, on a little wagon, where if you handle them at all, there's risk of breaking bones and so on. And I think the quality of life for these children has been dramatically impacted by the clinical trials he's done with these drugs.
We've been relatively good in the case of brittle bone disease, where we're making new diagnoses, new genetic causes for this group of conditions. And then now we are even treating them with therapies which can make the difference between walking and not walking in children.
Brendan's team is also focusing on a metabolic disorder in which eating protein-rich foods causes a chemical imbalance in the body.
And these red blood cells are very abnormal, and this is what happens often when there's an absence of nitric oxide in the body. The only reason we looked at this is because of our patients.
Brendan strives to keep his research patient-oriented. He founded the Skeletal Dysplasia Clinic at Texas Children's Hospital 9 years ago, where his clinical trials are helping patients who have run out of other options.
I think he's excited about what he's doing in terms of science, and he gets other people excited with him.
Seeing patients and taking care of patients, I think, is so inspiring. It's also a great source of ideas, because trying to figure out why someone is sick forces you to really think perhaps out of the box.
Sweet. So silicon nanocrystals in oxide.
In laboratories at the University of Texas at Austin, Dr. Brian Courville is leading a group of young chemical engineers. They're unlocking the secrets held by the universe and its elements.
So do you think you'll use some bismuth nanocrystals to make your...
As part of the Courville group, these graduate students are following in the footsteps of a man who has blazed a new trail in the study of nanomaterials and nanotechnology.
I love what I do because it's you're exploring into the unknown, and being in an academic world, you have quite a bit of freedom to discover and go off in different directions.
Brian's research focuses on the fact that the characteristics of some materials can be changed after they've been broken down into extremely small particles. Brian watches as this student uses high temperature and pressure to grow something called geranium nanowires. Another student is trying to change the known properties of silicon. Brian has been performing experiments like these for the past 10 years, and some of his work is a big reason the science is now taken seriously.
Well, I've observed Brian over the years, and he's one of the more creative young scientists that I've had the opportunity to work with and interact with. And I think what Brian brought to the campus and is his ability to work with nanoparticles in these very small structures and to really be out on the leading edge with respect to what their potential might be and in exploring new application areas for these materials.
For instance, what if the solar energy that's reflected off this building could be harvested by paint on its walls? Brian Courville and his students are trying to develop that technology. They're experimenting with elements that are capable of emitting light. The nanocrystals in these vials glow in different colors because they've been broken down to different sizes. Brian is also trying to develop nanowires that can revolutionize computers.
If you're a field general, maybe, or a general in the Army, you might want to have a display that rolls up like a map that you unfurl out on the battlefield and then is a moving computer screen. Nanowires are one aspect of what's needed to create that technology.
Someday, nanotechnology could also save lives. Brian and his students are trying to develop a synthesis that will change the properties of a nanomaterial so it can be safe enough to inject into the human body and unique enough to help doctors detect diseases like cancer.
We do a lot of fundamental discovery where we're learning what are the properties of materials, but then the engineering part of us, we're always trying to solve problems. So people are always coming to us with problems, and can you do this with a nanomaterial?
Brian Courville could use his vast knowledge of very small things to make large amounts of money in the private sector. But he says UT Austin is where he belongs.
So the science is really exciting and getting to discover things, and that's fantastic. But at the same time, interfacing with students and having an influence on some people's lives is pretty exciting also.
So this is some solution that we're gonna put that's gonna be going into the inside of the cell.
At UT Southwestern's Department of Pharmacology, Dr. Rama Ranganathan is shedding light on how proteins carry out vitally important chemical reactions in cells. Proteins are made up of amino acids, which seem to interact with each other like a tightly packed 3D jigsaw puzzle. Rama developed a new computational model revealing which amino acids are most critical for the structure and function of proteins.
We are looking at all the products, all the proteins that have survived over the ages through the process of evolution. And by looking at that ensemble of sequences from all the species, we can discern the basic rules by which the protein is made.
Rama's computational method showed the existence of tiny networks of amino acids that link distant sites on proteins. These networks allow proteins to communicate, carrying signals between different points like the wires in electrical circuits. Defects or mutations in these amino acid networks can trigger cells to behave improperly, causing diseases such as cancer or diabetes. Rama's computational method could potentially be applied to find out which sites in proteins are most sensitive to these mutations.
Understanding that allows us to begin to perhaps rationally design medicines or drugs that will alter that mutation so that it can now function perhaps.
Rama's model was new and different and initially had its skeptics.
Over 100 laboratories have taken this computational method that he's discovered and started to test them in their laboratories. And in every case that's been tested so far, it's stood the test.
Rama and his team more recently applied his computational method to an even more rigorous test, building a synthetic protein from scratch. Their result was several artificial proteins that showed the same structure and function as natural ones.
Being able to design a protein from scratch is I think, a significant step, because it means that the rules that we learned, we're on the right track.
Ultimately, these models for proteins have to be tested in living organisms to show that they can work in a natural setting. To do this, Rama and his team are working with bacteria, yeast, and flies to understand the ways natural and designed proteins may function differently in cells.
This is a fly that carries a mutant form of a protein.
Rama and other scientists are also testing his models for protein function using the neuronal cells in the eye that use a network of proteins to sense light. It's the first step in vision.
The neuron would then send the output out of this end of the cell to the rest of the brain.
Someday, the research taking place in Rama's lab could lead to new personalized medical strategies.
It's becoming evident that his work has the possibility to completely revolutionize how we understand protein architecture.
While Rama knows these goals are years down the road, he enjoys every discovery he makes along the way.
We have all these fantasies about big questions we want to address, but actually just the ability to contribute to these questions in some non-trivial way is already a success in my view.
Tonight, we honor 3 up-and-coming researchers, Brendan Lee, Brian Courville, and Rama Ranganathan, recipients of the 2009 Edith and Peter O'Donnell Awards.